This is component 3 mock feedback for paper 3. We're on to question 6, and we've got a smear of human blood, which just basically means a very thin sample of blood, not like a drop, but one that's been put onto a little drop and slide and then smeared across to make a very thin section so that you can actually see the actual cells. Label the components. Ooh, dear me. Right, well, I'm going to start off with one that I know. I'm going to have a red blood cell there. We can't say RBC because that could stand for absolutely anything. Or you could give it its nice posh site, erythrocyte. But I'll stick with red blood cell. Some other things, what do we know about the components of blood? Well, I know about white blood cells and plasma and platelets. Well, the plasma is this stuff, kind of a liquidy part around. Platelets are the small little things that help with clotting, so I'm going to go for this one here, platelet. And then I've got white blood cells, and you might, if you've done the immunity, then you'd know a little bit about white blood cells, or from GCSE, basically two types of white blood cells. White blood cells that produce antibodies and something to do the immune response that way, and other ones that kind of phagocytes that gobble up other things like bacteria and so on. But we haven't done those in a lot of detail, so all I can really say is white blood cell. And another type of white blood cell. There we go. State the main function of a cell labelled C. So that's the one that kind of looks like that, or side-on view would be our one like that. Main function of our red blood cell, it does contain, we say contains hemoglobin, but what's the actual function of it? It's to carry oxygen. So to carry oxygen or transport oxygen. And we could also say transport CO2 there, but the main function, oxygen. And we need an adaptation, so describe and explain with 2 marks. So we could do with the description over here, and then maybe the explanation over there. So describe and explain. These got to be linked together. So, for example, if we go for the obvious, we can have biconcave, which means it's concave on that side, and it's concave on the other side. And this increases the surface area. So that's one of our linked description to an explanation. And another thing that we know about, no nucleus, and so more space for hemoglobin. And there are some other things that we could say here. You don't need them, we've said 2 now, but we could talk about hemoglobin. So it's got hemoglobin, to transport oxygen. We could say it's thin to reduce the diffusion distance. We could say it's small. They are very small. Increase the surface area to volume ratio. But there's a couple that you should definitely thought of. And then part C, We don't know a lot about this, a little bit of general knowledge really, and stuff from GCSE that you remember. This is kind of aimed at, your A-star kind of candidates. And even if we lost these 4 marks, then you don't need 100 out of 100 to get an A-star. Generally, it might be, maybe about 80, who knows. Identification of the antigens present on the cell membranes is used to place people into different blood groups. What is meant by the term antigen? So we've got antigens, so these are on the cell membrane antigens there. Okay. And in blood we'd have A and B, and that's where we get our blood system from. What is meant by the term antigen? And I say it like that because it's, generates antibodies. That's the way I remember it. So it's an antibody generator, or will lead to that. But what actually is it? It's a protein. So it's a protein, or you could say, something on the cell surface. So it's a protein molecule, or a molecule on the cell surface. And this is for cell recognition and will elicit an immune response or stimulate antibody production. So it's a protein, and recognized as foreign, and will lead to antibodies being produced, will lead to antibodies being produced, or an immune response. And why it's important to identify a person's blood group correctly before giving that person a blood transfusion. Well, hopefully you're nearly at the state where you're donating blood. Well, the recipient's blood could contain antibodies against those antigens, like the A or the B, or there are some other blood groupings against these antigens. And this could cause all sorts of problems. For example, agglutination of red blood cells and antigen-antibody reaction. You might remember whatever from GCSE. So antibodies bind to the antigens. So antibodies would join onto these antigens and cause agglutination or attack by the white blood cells as a foreign. So I'm just going to write agglutination. But these, don't worry too much about these. It's just a little bit off spec. But certainly you can forgive yourself those, but certainly not carrying oxygen and shapes of red blood cells and so on. And that's 9 marks.